Photochemistry in detailed discussion for delayed fluorescence, IC inter-system crossing, IC internal conversion, and last topic that should be chemistry or vision for today's class. Okay. In previous class we studied detail about resonance fluorescence. Now we have to consider delayed fluorescence. What is delayed fluorescence? Okay. Now the delayed fluorescence there involves the process of inter-system crossing and also reverse inter-system crossing from T1 to S1. Meant there should be, for delayed fluorescence, that should be consisted of two inter-system crossing. One is inter-system crossing, other is reverse inter-system crossing. Okay. Meant there should be, when molecule or atoms they absorb some amount of energy, they get excited, and so they go ground state to excited state. That should be clear, right? Suppose they go S1, singlet excited state, then by non-radiative transition ISC they go T1. Okay, and again from T1 to S1 they again go. This process is termed as delayed fluorescence. Okay, so what you consider, there should be one ISC and there should be reverse ISC. Okay, that is S1 to go T1, again T1 to go S1. That should be termed as delayed fluorescence. Lifetime 10 to the power minus 4 second. 10 to the power minus 4 second. Delayed fluorescence result from two inter-system crossing. First, singlet to triplet. That should be singlet to triplet. That should be ISC. And there should be triplet to singlet. That is also ISC. Okay. One is singlet to triplet. That should be ISC. Others triplet to singlet. There should be ISC. Now properties, delayed fluorescence, which is also called delayed luminescence. Okay, we can say luminescence. Delayed fluorescence is which is also called delayed luminescence or delayed light emissions, or we can say afterglow. Is the extremely weak light emitted by P illuminated photosynthetic sample. Okay. We can say delayed fluorescence as the other name also, that should be delayed luminescence and delayed light emissions or afterglow. And that should be weak light emission emitted by P illuminated photosynthetic samples. Okay. The phenomena was discovered by Settler and Arnott in 1951, was related by them with process of photosynthesis. Delayed fluorescence is very much related to photosynthesis process. Okay. So delayed fluorescence is a two inter-system crossing process. There should be one singlet to triplet and triplet to singlet. Inter-system crossings are used. Okay. First on case singlet to triplet there should be inter-system crossing will be there, and triplet to singlet there should be reverse inter-system crossing will be there. And this combination are termed as delayed fluorescence. Okay. And that is a weak process that should be emitted by P illuminated photosynthetic samples. And we can also say delayed fluorescence as delayed luminescence, delayed light emissions or afterglow. Okay. Now, but there are delayed emissions whose spectra coincide exactly with the pumped fluorescence from the lowest signal state. The only difference is their lifetime. Okay. So for when molecule goes to first singlet excited state, on that case delayed fluorescence can be there and also fluorescence can be there. So sometimes pumped fluorescence that should be overlap with delayed fluorescence. So we cannot get so difference between of them. Okay. So there is only difference is their lifetime. The processes are known as delayed fluorescence. There should be only difference of between of them is their lifetime. Okay. The most important types of delayed fluorescence, basically two types. One is E-type, other is P-type. One is E-type delayed fluorescence, other is P-type delayed fluorescence. This is P-type. Okay. There should be molecule go H0 to S star. Okay. There should be pumped fluorescence. Okay. This should be internal conversion. There should be phosphorescence. Actually this should be straight line, this should be curved line. Okay. Phosphorescence, and there should be double ISC. Okay. Always remember, this absorption is a radiative process, so straight line. Fluorescence is a radiative process, so straight line, and phosphorescence, that is also a radiative process, straight line. And internal conversion and inter-system crossing will be the non-radiative process, so they are shown as curved line. Okay. Okay. Now consider in that place there should be double inter-system crossing will be take place. Okay. Okay. Now consider del EST greater than 0.4 electron volt. Okay. Del EST that should be greater than 0.4 electron volt, and there should be the process is actually, this can be give the definition of type P. That should be, there must be two triplet excited state produce one singlet excited state and one Gauss state. Okay. That is for P type. That is for P type. There should be two triplet excited state that combine to produce one singlet excited state and Gauss state. This should be termed as triplet annihilation. Okay. For type P delayed fluorescence, there should be two triplet excited state that should be produce one singlet excited state and one H0 gound singlet state. Okay. Now for E-type, that is also same. There should be absorption, there should be fluorescence, there should be phosphorescence. This is will be the straight line phosphorescence. This will be straight line. Okay. There should be double ISC. Okay. But there should be del ST less than 0.4 electron volt. They are greater than, there should be less than. Okay. There should be less than 0.4 electron volt. So del EST, by this way you can differentiate it. That is when type P, del EST that should be greater than 0.4 electron volt, and here for type E there should be del EST less than 0.4 electron volt. Okay. There this process is not to be applicable for type E-type process. E-type process there should be defined mechanism are seen. Okay. That is T1 star that should be produce S1 star and S0. This mechanism actually not applicable for type E. There should be another mechanism that should be used. Okay. Now we go inter-system crossing. In previous lecture, I briefly explained about inter-system crossing and IC. They are non-radiative process. Due to this transition, no radiation that should be evolved. Okay. Okay, inter-system crossing ISC in some molecules. there is a triplet state T1 which is close to an excited singlet state S1. So as a result, the molecule can cross over into triplet state such that of transition S1 to T1 is called as intersystem crossing IC. And it also a non-radiative transition, and the non-radiative transition do not emit any type of radiations. So as that is when molecule go S1, they are comparable energy, having comparable energy 1 T1 that should be present. Okay. So as their energy are almost comparable, or if not comparable, the energy difference is not so high. So on that case, S1 to T1 as well as sometimes T1 to S1 transition can be possible, and due to this transition no radiation that should be evolved. So that's why these are termed as non-radiative transition, and we can better say singlet to triplet and triplet to singlet transition are termed as intersystem crossing. Okay. This process involve transition between state of defined spin, that is spin multiplicity. As we know, for singlet there should be spin plus half. Okay, spin are not changed. Spin will be unchanged for singlet excited state. So that should be when in ground state S0. So when S1 S0 there should be this spin. S1 there should be one electron go in this ground state, other electron go excited state, and T1 triplet excited state there should be spin change. Okay, so by this analogy S1 equal to plus up and minus up, that is we get 0. So 2s plus 1 spin multiplicity will be 1. For singlet excited state S1 we get total spin equal to 0, and 2s plus 1 will be 1. That should be singlet. Okay, and for triplet we get total s will be 1 and 2s plus 1 will be 3. So spin multiplicity are defined. For S1 spin multiplicity is 1, for T1 spin multiplicity that should be 3. Okay. So from S1 to T2 and S1 to T1, these transitions are also called non-radiatory or radiationless because no radiation are removed. Okay, spectroscopically such transitions are forbidden because as we know, singlet to triplet and triplet to singlet transitions are symmetric forbidden. Okay, as spin that should be changed, and we know for selection rule always applicable, always favorable processes should maintain spin unchanged, del S equal to 0. But when triplet to singlet and singlet to triplet are transitions are occur, on that case spin are not unchanged, spin that should be changed, del S not equal to 0, and spin multiplicity that should be changed. So due to spin change, due to spin multiplicity change, and due to del S not equal to 0, we get symmetry forbidden or spin forbidden process. Okay. Now spectroscopically such transitions are forbidden, however they too occur though at relatively slow rate. As we know, due to two reasons, we can say that is not due to that is one reason, that should be due to two reasons. First one, there should be spin change. Okay, spin multiplicity change. So this is a slower process, less favorable, and always remember this process is forbidden process, and forbidden process are always slow process. Okay, so there should be S0 is ground state, S1 is a first excited singlet state, there should be intersystem crossing T1, S2 there should be intersystem crossing T2. Most of the cases S1 to T1 and S2 to T2 they are more favorable than reverse processes. Okay, because energy of S1 and T2 S2 that should be higher than T1 T2. So energy we do not need any energy have to be provided. Okay, so most of the books there should be provided ISC means S1 to T1, but revert can also be possible. Okay, so this is termed as reverse ISC. We can say reverse ISC, or may we use ISC term, but we can better use reverse ISC. And by these two combination, we know that is ISC and reverse ISC. This is called delayed fluorescence. Okay. Now we consider IC internal conversion. Internal conversion, that should be non-radiative and radiationless transition, and there should be occur due to singlet to singlet and triple to triple transition. As this is spin that should be unchanged, spin multiplicity that should be unchanged, del S equal to 0 maintain, that is symmetry allowed process. So spectroscopically these processes are allowed process. Okay. So the transition from excited singlet state S1 to ground singlet state S0, that is S1 to S0 are called non-radiative transition. We can say S2 to S1, also S3 to S2 also, they are also termed as IC. Okay, you can rectify it. S1 to S0, as we can say S2 to S1, S3 to S2, as well as T3 to T2, T2 to T1 transition are all called as intersystem crossing. It is also called intersystem crossing, and it can this is internal conversion. Okay, that is S3 to S2, S2 to S1, S1 to S0 sometimes can be occur, but is not always taken, and T2 to T3 to T2 and T2 to T1, they are termed as internal conversion. Okay, it can also take place between triplet state in the same manner, T2 to T1. Okay, from higher triplet state to the lower triplet state. Okay, as we know that is higher excited states are more energetic, so it must be more easy to remove energy and go lower excited state. Okay, that is more favorable. The transition involved the return of the activated molecule from the higher excited state, that is S3, S2 or T3, T2 to the first excited state. And this transition do not involve the emission of any radiation, that's why are termed as radiationless or non-radiative process. There should be some controversies, is written in many books. If you observe in photochemistry many books, there should be sometimes IC and intersystem crossing that should be given as that is T1 to S0 and S1 to S0, they are feasible. But many books there should be termed as this are not feasible, there should be only fluorescence and phosphorylation can be occur. So this controversy can be there in many sources. Okay, but we can use, there should be chance of internal conversion can be taken place in S1 to S0, and also intersystem crossing can be occur in T1 to S0, but these are rare. Okay, these are rare. Okay, we mainly use that is higher excited state, that should be S3 to S3 or S2 or T3 to T2 to S1 or T1. That should be S3 to S2, S2 to S1, T3 to T2 and T2 to T1. Okay. Now we consider chemistry of vision. Chemistry of vision. There should be H nu, that is suppose light that should be go on our eye. This is our eye. Okay, this is termed as retina. This is termed as fovea. There should be cones of color, cone cells are there. Okay, this is optical nerve, because optical nerve used for giving nerve impulses, that color or light that should be passes in our eye. On that case, optical nerve give the signal that yes, there should be some color or light that should be go on the eye. Okay, how we feel that color or light that should be passes in our eye, that is when they passes in our eye, optical nerve give the signal or impulse that light goes on our eyes. Okay, there should be retina, and retina having rod cells. Okay, retina having rod cells. that should be this. And fovea having cone cells, there should be colors are there. Okay, in there must be defined region, that is blue 450, green and red. Always remember, sensation give their corresponding their that blue give yellow, green give purple color, okay, red give bluish green color, like this sensation. Okay, now the question is, what is the role about beta carotene? Beta carotene found in more in carrot and less amount in pumpkin. Okay, that is, there should be in pumpkin, there must be two types of pigments are there. One is beta carotene, other is lycopene. Lycopene is found in greater extent, but in carrot there should be beta carotene present in major amount. So there should be beta carotene, there are huge number of double bonds are there. So due to this, there should be more conjugation. So color, that color is more intense. So that's why carrot color is orange. Okay, due to beta carotene. Now, when we taken beta carotene, that is 9 linear conjugated C double bond, that should be synthesized vitamin A, retinol. We can better say chemical name retinol. Okay, vitamin A. And this vitamin A oxidize in our body to produce this carbonyl, alcohol to carbonyl. Okay. Now, this compound react with lysine amino acid to produce this compound. Again, by cis trans isomerization, that should produce this, and ultimately we get source of impulse. Okay. Again, I described, that is, there should be beta carotene. We taken from any sources, carrot, pumpkin, or vitamin A tablets, so on. Suppose there should be beta carotene, we get. Suppose. So this beta carotene having 9 linear conjugated C double bond C group or C bond, and this vitamin A convert this beta carotene convert vitamin A. Okay, where terminal having alcohol. Okay, by oxidation this OH convert into double bond O. Okay, this double bond O react with lysine unit which having free NH2 group and produce this type of compound linkage. Now cis trans isomerization, we get this product. Okay, and ultimately we get source of impulse. That, yes, we see this thing. Okay, the compound responsible for human vision is element cis-retinal, which occurs in association with protein in the rod and cone shaped cell in the retina of the eye. So our human vision responsible for due to element cis-retinal, and which that should be associated with the protein which are present in rod and cone shaped cells in our retina of the eye. Okay, and when the vision light falls on the human eye, there should be photo excitation of the human eye. Okay, element cis-retinal takes place and result in the formation of element trans-retinal. There should be element cis-retinal convert into element trans-retinal, means cis-trans isolation can be take place. This that is element cis-retinal convert into element trans-retinal. Okay, and the free trans isomer is there enzymatically recovered to cis form again, which mistaken use recombines with the protein. Okay, which instantaneously recombines with the protein. Okay, and these molecular changes produce electrical potential. In our eye, the cis trans isomerization produce some electrical potential. Okay, which is transmitted to brain within 25 millionth of second. That is 25 10 to the power minus 6 second. Okay, this is termed as 25 million second after the flash of the color. Now the nerve impulse give the signal or impulse that yes, we see this. Okay, or we see the light. Okay, light passes in our eye. Okay, this is the concept of vision, chemistry of vision. Okay, now the summary is, this is first one, that is delayed fluorescence. Delayed fluorescence what? That is the two consecutive intersystem crossing. One is singlet to triplet intersystem crossing, other is triplet to singlet reverse intersystem crossing. Okay, and that should be lifetime 10 to the power minus 4 second. Properties, that should be weak. This is weak, extremely weak light emission by P illuminated organic samples or photosynthetic samples, and that should be discovered by Shetler and Arnott in 1951. That should be linked with photosynthesis, and we can say delayed fluorescence as delayed luminescence, delayed light emissions, or afterglow. Okay, these are basically two types. One is E type, other is P type. There must be E type del EST greater than 0.4 electron volt, and that the process, that is two triplet excited state, react to produce one singlet excited state and ground state. Other is E type, there should be less than 0.4 electron volt. Okay, now we go intersystem crossing. This is a transition of, that is triplet to singlet or singlet to triplet transition. More favorable, that is singlet to triplet, because energy of singlet is more. Okay, and that due to IC there should be no radiation can be revolved, so we can say intersystem crossing as a non-radiative transition. Okay, now internal conversion, there should be higher singlet excited state to lower singlet excited state, and lower and higher triplet excited state to lower triplet excited state. Energy transition, and there should be, due to transition, no radiation can be evolved, so we can say internal conversion as termed as, that is radiation-less or non-radiative process. There only transition can be there, upper excited state, upper singlet excited state to lower singlet excited state, or upper triplet excited state to lower triplet excited state. Now we consider chemistry of vision. There should be retina, there must be fovea having cone cells, and retina having rod cells, and they are responsible for vision. And optical nerve, that should be give the nerve impulse. Okay, so if we take in beta carotene, which having lots of C double bond C bond, and that should be converted into vitamin A. Vitamin A oxidized to produce terminal double bond O, and that should be react with L-lysine unit, and there should be cis trans isomerization. We get source of impulse. Now the last one, there should be our vision responsible due to 11 cis retinal, and that should be present as a protein in the rod and cone cells in retina of the eye. And there should be, when light go in our eye, there should be photo excitation, and due to this 11 cis retinal convert into 11 trans retinal, and due to this chemical changes, due to the structure changes, there should be the electrical potential generated, and this transmitted in our brain within 25 millionth of second, 25 in 10 to the power minus 5 second. So we get the color.